So I'm not a toxicologist, I'm a biologist. That's why we go sort of from initial toxicology type studies into more biological pathways such as inflammasome activation. So silica particles as well as asbestos fibers, they're very different physical-chemical properties, but yet they exert quite common pathological mechanisms. One of them is, of course, the highly important production of antioxidants, both particle as well as cell-derived, leading to epithelial damage and macrophage activation, heightening an inflammatory state, ultimately leading to fibroblast recruitment and activation, and overall leading to an imbalance between the extracellular matrix production and breakdown, resulting in fibrosis. So, quite common molecular mechanisms, although they result in two very distinct fibrotic diseases. So whereas silica exposure, depending a little bit on the type of setting in which people are exposed, so the old time quarry mining, for instance, after 20 to 30 years of exposure, whereas newer industries such as the denim sandblasting in 5 to 10 years will lead to the development of silicosis, with a very characteristic histopathological pattern characterized by these dense silicotic nodules, so whorled nodules mainly of collagen-rich material. Asbestos exposure, on the other hand, approximately same duration of exposure, not only leads to the well-known mesothelioma cancer type, but also to a fibrotic lung disease called asbestosis. And here the pathological characteristic is a much more diffuse pattern of pulmonary fibrosis in which asbestos bodies can be observed. So actually the remnants of the asbestos fibers and then a deposition of iron-rich material. So we were intrigued by the different outcomes of exposures, but yet quite similar pathogenic mechanisms. So in order to get more insight into the molecular patterns that are activated, we actually characterized the differential initial response, and we chose lung epithelial cells, not macrophages, as a primary target for the particles and the fibers. And we compared the response to crystalline silica on one hand and cristobalite asbestos on the other hand. Now, we did not use microgram doses in our experiment. We actually calculated the surface area of the particles and the fibers to actually be able to stimulate cells with equal surface area dosings, and we also checked that we were using equitoxic doses. So these are the characteristics of the silica particles and asbestos fibers that we used. So we're in the lower micrometer range. And this is just an electron micrograph of the fibers and the particles. So for our studies, we used an isolate of primary human bronchial epithelial cells. And as you can see here, we used 3 different concentrations based on the surface area characteristics of either cristobalite asbestos and cristobalite silica. And these doses are expressed as million square micrometers per square centimeters. And in the lower half, you can see the relative mass amounts, as you're most familiar looking at. So what you can appreciate is that at the higher doses, asbestos was more toxic compared to silica doses, equal surface area. But that the lower dose of asbestos and the 2 lower doses of silica caused cell death below 50% and no significant difference in toxicity. So these were the doses that we used for further experiments. So summarized in these dishes, representative of the conditions, we performed Affymetrix gene analysis. We analyzed these data by gene sifter analysis and then also with ingenuity pathway analysis. First of all, we performed unsupervised cluster analysis just to get a sense of if statistics is able to recognize the different treatments and the different dosing regimens. And this technique is indeed powerful enough not only to recognize the different types of material that the cells were exposed to, but also the different doses, with the lower dose of silica almost yielding the same level of gene expression as the control conditions. And this is the Venn diagram summarizing the number of gene changes and the commonalities between asbestos and silica. And as you can see, the lower dose of silica only resulted in 3 gene changes. There's about 200 genes that were commonly affected by silica and asbestos. There's a large set of genes specifically regulated by silica, and about 60 genes specifically affected by asbestos. So this is what the Ingenuity program analysis actually gives as a readout. So here we plotted actually the genes involved, the genes commonly altered by asbestos and silica into their biological pathways. What you can see up here is that one of those pathways commonly affected was glucocorticoid signaling, on which nothing much is known in both diseases. Secondly, as expected, there is a large group of genes, both cell surface receptors, common transcription factors such as NF-kappa B and AP1, and then a myriad of pro-inflammatory cytokines and chemokines involved in leukocyte migration and inflammation. We also see a number of genes involved in apoptosis regulation, as well as cellular stress response, with a number of heat shock proteins being upregulated. And lastly, also not quite unexpectedly, genes involved in extracellular matrix dissolution, as well as fibrotic responses. So when we look at the genes that were specifically affected by asbestos, most of these genes upregulated, we read cancer all over the pathways. So specific cancer genes, but also genes involved in proliferation, regulation of cell death and survival, tumor morphology and remodeling, and also a group of genes involved in detoxification, amongst which the sulfotransferases and CYP proteins. Silica-specific alterations, maybe it's because the dose was slightly higher surface area-wise compared to asbestos, but on top of all the common gene changes involved in inflammation, there is many, many more that are induced specifically by silica. Again, a lot of cell surface receptors, some of which involved in pattern recognition, which is quite interesting because it's not a biological molecule from a sense of what we know, protein recognition, for instance. An additional group of inflammatory mediators, and I'd like to also point out alternative NF-kappa B signaling as opposed to common activation of canonical. Also quite intriguing is a marked interferon response to silica particles. And again, genes involved in cell survival regulation with a downregulation of a number of BCL proteins and fibrogenesis-related proteins. And here I'd like to point out that we see Wnt signaling pathway could be affected specifically by silica, which we have investigated further, but which I won't go into detail in this presentation. So, on to the second half of the presentation. One of the pathways we noticed to be upregulated both by asbestos and silica, and not only in primary bronchial epithelial cells, but also in an immortalized cell line, BS2B, was the inflammasome. Now, everybody knows toll-like receptors as being mostly cell-surface located pathogenic recognition receptors. But there's also a class of intracellular damage-associated molecular pattern receptors, and one of those proteins is the inflammasome. Most investigated is the NLRP3 protein. So this protein by itself is not sufficient to cause any effect. Upon recognition of a danger, here the agonist, NLRP3 needs to recruit an adaptor protein ASC, and that in turn leads to the activation of pro-caspase-1 in this multi-protein complex. And active caspase 1 is then able to release and activate highly pyrogenic cytokines IL-1 beta as well as IL-18. Importantly, inflammasome activation is not only associated with the release of IL-1 and IL-18, it's also involved in mediating a pro-inflammatory form of cell death, through which additional molecules are released from cells, such as HMGB1. Now, the inflammasome, you can imagine, has been widely studied in cells of the innate immune system, as that's where its function would be logical. And in these cells, mechanisms for activation have been described. in which mostly a priming signal is first required through a TLR4 mediated mechanism, leading to actually increased levels of NLRP3 as well as pro-IL-1 beta and pro-IL-18, as these levels are normally kept low in these cells to avoid overactivation of the system. A second activating signal for the inflammasome itself then leads to the activation of caspase-1. And it's not the damage that is recognized that is activating the caspase and the NLRP3 itself. It's actually intermediate molecular processes, such as, for instance, the release of ATP, oxidative damage to mitochondria, or lysosomal disruption, which are actually sensed by the inflammasome. Now, in 2008 already, silica particles as well as asbestos were one of the first particles to actually be recognized to activate this inflammasome. 3 separate labs performed these studies, and they focused again mostly on the innate immune cells. What they also went on to show is that mice deficient in the adaptor protein ASP as well as in the NLRP3 itself were protected against silicosis-induced inflammation, as well as silica-induced remodeling here in the bottom panel as shown by collagen deposition. So this is the actual gene list, genes associated with the NLRP3 inflammasome that we found to be mostly highly upregulated in both cell types in our experiments, so which prompted us to investigate the role of the NLRP3 in lung epithelial cells. So here we confirmed actually the microarray findings in that both asbestos and silica were able to activate caspase-1 activity to a very similar extent at equal surface areas. We also confirmed that this is specific to silica, and glass beads, which we included as a negative control in this experiment, were not able to induce the formation of active caspase subunits. Then for readouts, we assessed the release of IL-1 beta in response to silica. And what you can see here is that indeed silica treatment by itself is able to induce significant amounts of IL-1 beta. Significant in a sense, if you were here for the first talk, statistically significantly more IL-1 beta, but we are well aware that the levels of IL-1 beta released by epithelial cells is quite low. LPS is not necessary as a priming signal as opposed to the results proven in innate immune cells. And we show also that this is dependent on the presence of NLRP3, as siRNA-mediated knockdown of NLRP3, silica is no longer able to induce IL-1 beta release. We see the same, we'll go over it more quickly, for the release of BFGF. So induction of BFGF release in response to silica, there's no need for initial priming, and this is NLRP3 dependent. And also the release of the danger molecule HMGB1 can be evoked by silica treatment alone, and this is partially NLRP3 dependent. We then also assessed whether the particles really need to enter the cell in order to activate the inflammasome, and we assessed this by blocking first the uptake of the particles by cytochalasin D treatment. So in the presence of cytochalasin D pre-treatment for 30 minutes, silica is no longer able to induce the release of BFGF. And we see similar results for the release of HMGB1. So what does this all mean? As I said, the levels of, for instance, IL-1 beta are sort of meaningless, albeit increased after silica treatment. So we actually investigated paracrine signaling in an in vitro system. So again, BS2B cells were either pre-treated with cytochalasin D to block the particle uptake, or we knocked out NLRP3 and treated these bronchial epithelial cells with silica. After 24 hours, we recovered the cell medium and removed the silica particles, and we transferred this particle-free medium to an MRC5 fibroblast cell line. And here we assessed the growth of these fibroblasts by DNA content. These are the results for the treatment with cytochalasin D. So if we take medium from silica-treated bronchial epithelial cells we see a marked effect on fibroblast proliferation. However, if the epithelial cells were treated with cytochalasin D, so they were not able to take up the silica particles, then the media does not really induce any fibroblast proliferation. And in this experiment, we show that this is also dependent on the initial NLRP3 presence in the epithelial cells, as knockout of NLRP3 by siRNA also blunts the proliferative capacity of the BS2B medium. Another question we asked in a follow-up study was, does the particle reactivity matter? So they have to enter the cell, but do you need to have a reactive particle? From our glass bead experiments, we could argue that it doesn't. So for this study, we collaborated with Holzkens in Düsseldorf and compared our initial studies with cristobalite silica with Dorndorfer quartz 12 silica particles, because there we had access to DQ12 that was coated with a PVNO polymer, so to sort of scavenge all the surface reactivity. So here is again the IL-1 beta released by the bronchial epithelial cells, and we see that both cristobalite silica as well as DQ12 silica lead to about the same levels of IL-1 beta release. But if the DQ12 particle surface reactivity is scavenged by the PVNO coating, IL-1 beta release is no longer induced. And we see similar results for the release of BFGF dependent on the surface reactivity of the particles, as well as HMGD1. And both polymers of silica behaved quite similarly, again, at equal surface area doses. We also had access to an in vivo study in which rats were either instilled with PBS as a control, native DQ12, or the DQ12 that was coated with the PVNO polymer. And we also assessed inflammasome activation in this rat model used to working with mice. So this is an immunoblot of the BAL fluid, so the lavage fluid recovered from the lungs, and the animals were either sacrificed 3 days after the installation of DQ12 PVNO, or 7 days post the installation. And here again you can see the pro-caspase-1 as well as active subunits, and the DQ12 treated rats, there is an increase in active caspase subunits in the BAL fluid, which is attenuated when the PVNO coated quartz was instilled, which is quite remarkable that this caspase activity is not just inside the cell, but can also be released. So we also assessed where inflammasome activation is located, because as I said, most studies focus on the myeloid cells. And as you can see in blue here, we stained for caspase-1, and in the right-hand panels we stained for IL-1 beta. So this is 180 days after the installation of the particles. We still see a marked upregulation of both caspase-1 and IL-1 beta blue staining, and not only in the inflammatory cells that have infiltrated the lung tissue, but also in bronchial epithelial as well as alveolar epithelial cells. And this heightened presence of caspase-1 and IL-1 beta is markedly attenuated when the rats were instilled with PVNO-coated particles. So this still doesn't get to the mechanism as how silica particles activate the inflammasome, because I said it's not a paradigm that the particles interact with the inflammasome and directly mediate its assembly and activation. One key molecule known to be linked to inflammasome activation coupled to redox stress is TXNIP. And TXNIP is thyrodoxin interacting protein, but its original name was actually vitamin D binding protein. So under normal resting circumstances, TXNIP is a binding partner to the antioxidant thyrodoxin. But upon oxidative stress, thyrodoxin is oxidized, and thereby TXNIP can be released from its binding partner, and it's believed that TXNIP is the actual protein that couples to the NLRP3 inflammasome and leads to its activation. Now, some studies have manipulated TXNIP levels to prove that this is a way that the inflammasome is activated, but we went about more the pharmacological approach, in that we tried to increase thyrodoxin levels, so to sort of scavenge and keep quiet the thyrodoxin, keep it bound. So in this upper set of experiments, we actually overexpressed human thyroidoxin, and we again stimulated the cells with cristobalite silica, and we looked at IL-1 beta and HMGB1 release. And as you can see, under control vector conditions, silica is able to induce IL-1 beta as well as HMGB1 release. But when we overexpress thyroidoxin, this response is blunted or completely absent. A similar approach is actually to treat the cells with recombinant human thyroidoxin, which is proven in many animal studies to be protective against multiple inflammatory diseases. And similar results can be obtained. So addition of recombinant thyroidoxin also prevents the activation of the inflammasome and the release of IL-1 and HMGB1. So sort of in summary for this inflammasome work, we show that treatment of epithelial cells with silica particles leads to the activation of the inflammasome and the release of important mediators such as HMGB1, BFGF, and IL-1 beta. In aggregate, this conditioned media of these cells leads to increased fibroblast proliferation. On the mechanistic side, we show that the activation of the inflammasome is uptake dependent and also dependent on particle surface reactivity. And we also show an important role for the TXNIP thyroidoxin axis in activating this pathway. Now, this isn't just basic biology, this isn't just screening. Earlier this year in a reputed lung journal, there was actually a case report. It's about a 37-year-old male marble cave worker who developed silicosis, and the doctors were sort of at a loss of how to treat the patient, how to make him better, sort of were giving up. So what they did is they actually put the person on 6 months of an anakinra treatment, and anakinra is a commercially available IL-1 receptor antagonist, so it is a mediator of inflammasome activity. And I'm not a toxicologist, but I am not a pulmonologist either, so forgive me that I don't know exactly how to interpret these scan results. But I think what everybody can appreciate is that at least PaO2 was drastically increased after this 6-month regimen. And also the disease activity was markedly lower. It's an N of 1, but it's at least promising that a target in which we are studying in a biological system can actually make it to the hospital, to the bedside. So with this, I would like to acknowledge the most important people who actually performed this research, and those are my 2 PhD students, Paul Peters, who recently received his PhD, and Timothy Perkins, who will do so in the near future. Our collaborators at the University of Vermont, specifically Brooke Mossman, who was very instrumental in setting up these studies, and as I mentioned, collaborators in Düsseldorf for the DQ12 studies. Thank you.
Thank you, Dr. Reynaert, for that fine presentation. Are there any questions? But can I stop asking, you clearly looked at, in vivo at least, looked at the rat model.
Mm-hmm.
Are you planning to looking at any other species? I mean, rat model, how does it bear up?
Well,
versus humans?
Versus humans, of course, it doesn't really bear up. We have actually also set a collaboration with Bochum, where they have access to old autopsy material of miners. So we have actually also stained for these inflammasome readouts in miners who developed silicosis versus miners who had equal exposure times, equal exposure, estimated exposures, who did not develop silicosis. And we do see differences, but it's very hard to quantify these differences, and also because the tissue looks so much different. And a non-silicotic miner does not have normal looking lung tissue. And then in the silicotic nodules, they're absent in the non-silicotic. So we have done those studies, but at least we see presence of these mediators at time of death. So it must mean something.
Okay, good. I mean, how do you translate what you see in to taking that further in terms of application where findings have in improving human health, particularly obviously?
Well, I think there's a couple of issues there. I mean, the last slide I showed is that therapeutically it can be translated into the clinic. Another means would be to actually look at screening, so looking at these inflammasome readouts as a method of screening, so to identify the at-risk population versus the non-at-risk. And there it's actually not just the readouts, but also potential single nucleotide polymorphisms in some of the genes that code for these pathways that, especially in China, have been shown to be linked to actually prone to disease development. So that's one other approach. But I think it also goes back to, is there really a disconnect between inflammation and the development of fibrosis? If the inflammasome can be targeted after fibrosis has already been established and it can improve quality of life, inflammation, lung function, then there is also some means of saying that they are not just disconnected as some studies suggest.
I was wondering about that case study. Did they talk about symptomology? Like, did it improve his quality of life?
Besides this very short list, and they've also mentioned inflammatory markers going down without actually putting in the numbers, but they did say that the patient felt better and was doing better. I mean, yeah, it's really strange to be presenting an N of 1, but it's so promising, so hopeful for a large group of patients that is still increasing. It's not a historic disease by any means.
Picking up from as the further reporting of the same patient?
Well, this was just earlier this year, so I don't know. But pharma is all over this pathway, so for asthma, for COPD, that's just lung that I know of. But it's a very hot pathway.
Thanks ahead.
Yeah.
Good. Thank you very much.